And this kind of movement we call the continuous conduction. And let's look at the saltatory conduction. In some axons, especially the one need to travel for long distance, like the motor neuron, they go from the spinal cord, go all the way to control your finger, control your toe, and this axon can be 1 meter long. And you want the signal to travel for long distance, they create the myelin. The myelin is created by the glia cells. They are not neurons, they are like supporting unit. If you think about the neurons are the combat unit in military, these glia cells are supporting unit. So we have 6 different kind of glia cells. If you took anatomy, you learn all of them. Right? So the one create a myelin in the central nervous system is called oligodendrocyte. In the peripheral nervous system is called the Schwann cell. Apparently Schwann is the scientist who found it. And these myelin are like insulation. They cover 90% of the axon. It's like you have an electrical wire, and now they cover 90% of it. All you need to worry about is those exposed one. They call the node of Ranvier. Ranvier is the person who found this. Node of Ranvier is those exposed one. So apparently all those voltage-gated ion channels, they are located in the node of Ranvier only. And the first action potential is here. Next one, they don't have to worry about the myelinated one. They cover 90% of them. So now the next one is not here, it's here. It's like they jump. They call it saltatory conduction. It's like jumping. If you think about you walk from the building, you're located to the UT main building, UT tower. If I ask you to walk heel to toe, that's the continuous movement. It's gonna take a while for you to reach there. You would rather you can jump or you can run, right? And that's the saltatory conduction. And especially for the neurons, signal travel for long distance, like the motor neuron. Pretty much all of them have the myelin, so they can use the saltatory conduction and make sure the signal can travel and can go faster. If you damage this, sometimes the signal can, sometimes the signal could not move out, and what's going to happen is the motor control problem. So we have a disease called multiple sclerosis. It happens, it's an autoimmune disease. That's the immune system, and they see those myelins and they say they look suspicious. Let's kill them. So it turns out these are the myelin cells, and when you don't have myelin, the signals may not go to the end part and reach its target. Target is skeletal muscle. So in the multiple sclerosis, their symptom is they lose the motor control. They lose the motor control. So even though the problem comes from the neuron, so it's a neuron disease, it's autoimmune, so it's the immune system. So far no treatments. It's the immune system. Then most of the time they make good judgment. They fight bacteria, then leave the good cell alone. And sometimes they make the wrong decision. So they damage the oligodendrocyte. It turns out it's multiple sclerosis. And we also have other kind of autoimmune disease, like we talk about diabetes 1. There's the immune system attack the beta cells in the pancreas. So, and the beta cells produce insulin. So diabetes 1 patient have no insulin. They have to self-inject insulin. It's another autoimmune disease. And when they attack this, the symptom is you lose the motor control. So multiple sclerosis patient, they gradually lose their motor control. And so far no treatment. They can only slow down the process. They could not reverse it. Okay, so that's the action potential. And this picture put the action potential, this is the red line, together with the permeability of sodium and permeability of potassium. Because in the axon, you have the voltage-gated sodium channel and the voltage-gated potassium channel. You found the action potential, especially in the depolarization part, it overlapped with the sodium one. This tells you in the depolarization part is due to sodium. It's due to sodium. It's the opening of the voltage-gated sodium channel make the quick depolarization of the action potential. And the voltage-gated ion channel, they open, then they quickly close. So they quickly close. When they close, and the action potential start to have repolarization. And also, that's the time the voltage-gated potassium channel open. it's still very quick from here to here, take about 1 to 2 milliseconds. Voltage-gated sodium, voltage-gated potassium, they both sense the voltage, and they both open. But sodium channel is a little bit quicker than the potassium channel, 1 millisecond quicker. It's like if you watch the fencing, wow, they're super fast. And in Olympic, and they just, both the athletes are super good, super fast, but one of them win. It turned out this one voltage-gated sodium channel just 1 millisecond faster than the voltage-gated potassium channel. So when the sodium channel open, potassium channel haven't opened yet, so it causes the depolarization, sodium flow in. Then sodium channel close, and potassium channel open. So the repolarization part is mainly caused by the voltage-gated potassium channel. And now finish one action potential. Take about 2 to 3 milliseconds. And from here to here, that's the time this voltage-gated sodium and voltage-gated potassium channel are super busy. They're working on this action potential. They don't have time to do something else. So the neuron won't be able to generate another action potential. And we call this period of time refractory period. So refractory period is a time you won't be able to generate another action potential. So in neuron, it's very quick. From here to here, 3-4 milliseconds, you can generate another one. And when we talk about the heart muscle, when we talk about EKG, you will see I use this term again, refractory period. In the cardiac muscle, refractory period is very long. It takes about 200 milliseconds. It's very long. It's a protective mechanism. So it makes the heart muscle won't be able to generate another action potential after 200 milliseconds. So it's a protective mechanism. It prevents your heart rate go to 500 beats per minute. So you found your heart rate is about 150, 160. It won't go to maximally 200 beats per minute. That's the maximum. And the reason is the refractory period is long. So refractory period is the time you won't be able to generate another action potential. This is called the refractory period. And now let's look at what's the absolute refractory, relative refractory. That happens in a lab. So those electrophysiologists in the lab, they can make the neuron generate action potential. They can provide stimulus to force them to generate another action potential. In the axon, you have so many voltage-gated ion channels. Once they open, and now let's look at why they have the refractory period. They have a gate. They also have a plug. So after the gate open, the plug quickly plug in. So that's why they are so efficient. Generate the depolarization and stop, because the plug in. Once they plug in, there's no way the ions can go through. And you have the one more for the potassium as well, right? They also have a plug. Okay, once they plug in, it takes a while for them to unplug and the gate close. And say you have 1,000 voltage-gated sodium channel, not all of them unplug at the same time. One unplug a little bit faster than the other. Say this guy unplug 1 millisecond faster than the other. And once you have enough voltage-gated sodium channel being unplugged, say you have 1,000 of them, once you have 600 of them being unplugged, you can force them to generate another action potential, even though this won't naturally happen. But when you give them a bigger stimulus than normal, so this happens in a lab, you can force them to generate another one. This is called the relative refractory period. So the neurons, usually they take about 3-4 milliseconds to generate action potential, but if you want to push them to their limit, they can generate action potential in 2 milliseconds, because their absolute refractory period is just 2 milliseconds. So your neuron can fire like crazy, generate action potential like crazy. They can generate a lot. They also produce a lot of ATP, a lot of waste particles. So you have glial cells remove those waste particles. If you block the blood flow to the brain, you found after 3 minutes the neurons start die, and that's stroke, ischemic stroke. That's when you block the blood flow to the brain, and the neurons start to die because they use a lot of energy. Okay, so we start from the dendrite and graded potential, and now the signal go to the axon here lock, generate action potential, and go to the saltatory conduction or continuous conduction. Now the signal reach the terminal. The axon terminal, that's the area this neuron innervate. Innervate is a big word to say that the neuron touch the other neuron, innervate the other neuron. So in this area, they're gonna innervate the other neuron, and this area called synapse. Synapse, it looks like they physically touch, but actually they don't physically touch. There's a small gap. So this is the neuron A, and this is neuron B. So there's a very small gap called synaptic cleft. This cleft is nanometer range. It's 10 to minus 10, 10 to minus 9 meter. It's nanometer range. Very, very small gap. So, but a small gap is still a gap. And the electrical signal start from the neuron A won't be able to go through the gap to neuron B. So in this area, the electrical signal need to transfer into chemical signal. So in your brain, you have two kinds of signal. You have electrical signal, you have the chemical signal. And the electrical signal, chemical signal, both of them need to be balanced. Electrical signal, if you have too big, there's a disease called epilepsy. And now let's look at the chemical signal. Chemical signal, that's where the neurotransmitter release happens. And they also need to be balanced. If you don't have enough, there's a lot of target of pharmaceutical medicine. When you don't have enough serotonin, it affects your mood. So you found antidepressant, they try to increase the serotonin level in this. That's when they don't have enough. And when you have too much, there's also a problem. Like people do drugs, and they feel so high because they have way too much neurotransmitter here, and they feel pretty high. But there's always a side effect. So now you gradually build a concept in your body. Everything need to be just right. So when you have too much, eventually you cause the withdrawal effect. Your brain put less receptor here, try to protect you, and when you release normal neurotransmitter, and you won't trigger the same response. So in those people experience drug withdrawal, they have those dual response. They don't feel anything can excite them in their life because they don't have enough receptor. Okay, so this area is a big target for pharmaceutical medicine. So let's see what's the mechanism to trigger neurotransmitter release. That's the chemical signal. And in the synapse, that's the areas you need to have neurotransmitter. And neurotransmitter they were packed in the vesicle. So you found they're like peptide hormone. They're not like lipid hormone, because lipid hormone, they are made by demand, right? They have to release out. And these neurotransmitters, they can be packed in the vesicle and ready to be released. So they're made of peptide. They're like peptide hormone. And in this area, a lot of membrane protein got involved. So if you took molecular biology, they were going to spend 2 weeks talk about the membrane protein got involved here. And here we focus on the ion channel. So the ion channel in the synapse area trigger the vesicle release is voltage-gated calcium channel. So that's the calcium. Calcium, when we talk about the second messenger, I told you the calcium is also a second messenger. And it works here in neuron and also in muscle. Calcium also triggers the muscle contraction. And here the calcium triggers the neurotransmitter release. So when the action potential goes through the whole axon, reach the axon terminal, it will open the voltage-gated calcium channel, of course, because the depolarization reach here. And calcium is high outside, low inside. Inside we also almost have no calcium. You can assume the calcium concentration is almost zero inside, because calcium is the second messenger. So once you have calcium, they will do something. So the inside has almost no calcium. And when you open the voltage-gated calcium channel, calcium flow in, calcium will trigger the fusion of the vesicle. And that's exocytosis. So exocytosis is releasing out. It happens in neuron, happens in endocrine cells. And when they fuse, okay, this neurotransmitter simple diffusion, and from here to here, they can bind with the receptor. So they transfer the electrical signal to the chemical signal. Okay, let's take a break.